Who is Erin Dueber? Erin Dueber is a biophysicist who researches drug discovery, specifically how macromolecules and DNA interact with certain substances. Erin Dueber grew up in Alaska. While growing up in Alaska, she grew an appreciation for nature before diving into scientific research in high school. After her research, she decided to pursue it as a career. She is now a senior principal scientist and group leader in the Department of Early Discovery Biochemistry at Genentech. Dueber always appreciated the nature around her from a young age, but never was interested in science until high school, where she learned about DNA proteins. Living near a college town, Dueber had the opportunity to experience a summer research internship at the University of Alaska Fairbanks. After completing this program, she solidified her decision in pursuing research. Dueber started her journey at Gustavus Adolphus College in Minnesota, where she double majored in biochemistry and chemistry. She then attended University of California, San Francisco, where she warranted her PhD in the lab of David Agard in the biophysics program. In her following years, Dueber worked as an independent postdoctoral fellow at the Miller Institute for Basic Research at the University of California, Berkeley. Her work at Berkeley was focused on X-ray crystallography of DNA protein complexes in RNA replication initiation. Dueber's biggest challenge was establishing her career and simultaneously starting a family. She learned to ask for help and be adaptable. Dueber says if she weren't a scientist, she would pursue investigative journalism, though she would not enjoy the deadlines. Dueber also likes to bake, and those close to her told her she should open a bakery. Though she said she's done enough school bake sales to know this hobby is not fit for a career. I admire Erin Dueber because she solidified her decision to become a scientist at a young age and stuck with it, even though it might have been hard. I've always wanted to become a pharmaceutical scientist since the 8th grade. She established a name for herself and works for a multi-billion-dollar company. That is my dream someday. She and I are pretty similar. If I weren't pursuing science, I would also pursue journalism because I do love writing. I think I could be a scientific journalist, though I would also not enjoy the deadlines. I had the opportunity to visit Genentech, the company Dueber works for, and is also how I found Dueber, and I fell in love with it. I left that day feeling so hopeful and excited for the future. Because of that very special and unique experience, I considered going to school in San Francisco and applied. Though I was accepted and awarded scholarships, it was ultimately too expensive for now, but I've been thinking of going to San Francisco for grad school, which is exactly what Erin Dueber did. Dueber is specifically interested in proteins and macromolecules, topics such as phosphorylation and proteolysis. Her focus is drug discovery. Dueber is all things macromolecule and DNA focused. Her big question is, what are the effects of specific enzymes in regards to proteins? A recent article by Dueber, Phosphorylation and Linear Ubiquitin Direct A20 Inhibition of Inflammation. Several experiments were performed to investigate how phosphorylation and linear ubiquitination relate A20's role in inhibiting inflammation. Western blotting was used to examine phosphorylation and ubiquitination of proteins involved in inflammation, and mouse models were used to assess the in vivo effects of A20 on inflammation. The techniques used in this study, including cell-based assays, protein interaction studies, and animal models, were essential to generate the figures that depicted the molecular mechanisms of A20 regulation and inflammation. In this figure, Figure 5, focuses on demonstrating the role of A20 in regulating inflammation through its interaction with linear ubiquitin chains and its phosphorylation status. The main conclusion from this figure is that phosphorylation of A20 enhances its ability to bind to linear ubiquitin chains, which in turn increases A20's capacity to inhibit NF-kappa B signaling. So who is Erin Dueber? She is a biophysicist from Alaska who performs research at Genentech who focuses on macromolecules and proteins.